Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. In an examine series content piece, we're going to be discussing a paper that's related to cancer. So we're going to go and introduce the topic, then we're going to go through and look at all the figures from this paper, and then we're going to finish this off with a conclusion. What can we take away from the paper? If you're interested, because this is an open source paper, I can supply the paper, which will be linked down below, so you can click on it and follow along and make your own notes and make your own interpretations. I strongly encourage you to comment and let me know your thoughts on this format, but also let me know what you think of the paper. What would you agree with? What would you not agree with? So in a short, sweet, science-based video, let's jump right into it. So what is this paper on? Generally, just to introduce it, this is, I may have mentioned it already, but a cancer paper. So we're going to be investigating how cancer can migrate through what's called our extracellular matrix. So although you have your cells, on the outside of your cells there are different collagen fibers and different fibers in general that make up a structure, and that structure allows for, well, just generally holding things together in a particular environment. So how does cancer move through that environment, especially if it is pretty rigid? I mean, collagen is relatively tough. It's not that bendable. It's got quite some rigidity to it. So how is a cancer cell, able to move through that collagen and move through our connective tissue to metastasize and eventually find its way to our bloodstream and then metastasize to a different area of the body? Now, this is a situation in which cancer has started to grow in a focal area, so in a singular area, and has started migrating outside of that area, but has not quite achieved metastasis to a point where it has moved into the bloodstream and then moved to a different area of the body. So it's essentially this precursor situation where it's trying to move to a different place, but to do that, it has to be able to move through our connective tissue. So this paper investigates exactly how it does that, and there are certainly more ways than just these ways, but there are what's called metalloproteinases or metalloproteases that are found on the cellular membrane of the actual cancer cell, and there are also these MMPs, which is just the shortened version of the metalloproteinase. I'll probably use them interchangeably. And these MMPs can either be secreted, as in released from the cell to attack collagen fibers, or they can be hunkered down in the actual membrane of the cell and have direct contact with collagen. So we're going to be discussing essentially three different types of MMPs, but we're going to be categorizing them into two different categories. So one is the secreted style, which is MMP1 and MMP2, which are in the paper. And then we have our MT1 MMPs. So the MT1 is the one that's bound to the actual membrane itself. Now the paper is trying to answer the question of which MMPs are necessary for cancer to progress through our extracellular matrix. And not only that, what happens if we knock out those MMPs? If we are able to inhibit the action of those MMPs, can cancer still progress? So, without further ado, let's go ahead and jump into the first figure. Okay, so let's dive right into figure one. And just to make sure that we're on the same page, so you know that the MMPs are the metalloproteinases or proteases that degrade collagen, because collagen is made up of, well, three different molecules, and they're made up of different amino acids. So these proteases are going to break up these amino acids and liberate them, so free them up, essentially breaking apart the fiber of collagen. So these MMPs, in this situation, we have three different MMPs, but they're broken up into two different categories. a secretory system, and a membrane-bound system. So the MT1s are the ones that are membrane-bound, and the MMPs 1 and 2, which are the two others, those are in the secretory system. So for figure 1, what they've done is they've put cancer cells, specifically HT1080s, which are fibrosarcomas, and they also have another line, which is MDA-MB231, and those are a breast cancer line, a highly aggressive triple negative breast cancer line. What that means, if it's triple negative, is essentially, I'm not going to bog you down with everything, but that it's incredibly aggressive, so it'll metastasize quickly. What they're looking at here is just, are you going to get cell migration? Are you going to get cancer migration if you try and knock down these particular MMPs? And the way they do that is by putting cells in kind of a spherical, like a droplet essentially, and then they put that on a collagen matrix, so the actual environment that they're trying to investigate, and then they watch, or they wait a few days, and they see if the cells migrate. So what they're actually also doing, if you'll notice with the MMP-1,2 siRNA, an siRNA is a silencing RNA, essentially binds to RNA and stops the translation of that messenger RNA to a protein. So it essentially knocks down the expression of that particular MMP in this situation. So these two MMPs are being knocked down. That's essentially the point I'm trying to get across here. So they've got their control, as you can see on the left. I'm not going to touch on that, but so for the MMP-1,2 siRNA, you can see that even though they knock it down, there's still cell migration. So the cancer cells are spreading across the collagen. Now in the MT1 scenario, however, they're knocking that down, and what you're seeing is that there's no cell migration, or very little cell migration. So in that situation, you're seeing that knocking down MT1, that particular MMP, that particular metalloproteinase, is incredibly effective. And then when they rescue it, so essentially they knock it down and then they reintroduce it, they find that it reintroduces the ability for those cells to migrate again. And the exact same thing is seen for the other cell line, the MDA-MB231, but in this situation, they're also adding a general inhibitor of MMP. So I'm not exactly sure how it works, but it does inhibit the action of those MMPs. So those MMPs can no longer degrade the collagen, and that's going to affect the MMP-1, 2, and the MT1. And what you find clearly is that there's no cell migration with that inhibition. The next one's B. All they're doing for B is they're trying to show that their silencing RNA works, and clearly it does. That's really all I'm going to touch on that. For C, they're essentially measuring the distance at which these cells are migrating. So how far are they migrating? And it's essentially non-significant for all three cell lines. They added a cell line here. Most likely the reviewers asked them to add another cancer cell line. The SUM159 is another breast cancer cell line, so it's another triple negative breast cancer cell line. And they found the only difference was, and this is consistent across all three cell lines, is that if you knock down MT1, then you get no cell migration. So that's essentially the point that they're trying to get across, and that's pretty poignant in its own right. And that's all I'm going to touch on for this first figure, mainly because I don't want to go over every single panel because a lot of it can get repetitive, but that's it for the first figure. Okay, for figure 2, what they're doing is they're changing up the situation that the cancer cells are encountering. So I'm not going to cover A mainly because it's essentially covered in figure 2B. So in figure 2B, you're seeing that there are two different types of collagen that they're using. So on a collagen matrix that they're putting the cells on, they've got pepsin extracted, which essentially means that the proteins that make up the collagen are completely broken up into their polypeptide chain. So they're no longer linked together, but they're also no longer, they're essentially degraded down to their constituent parts. So you can see that the cell migration is considerably different with the pepsin extracted collagen as opposed to the other form that they used is a reduced collagen. So what they added was sodium borohydride, and that essentially removes the cross-links between different collagen fibers. If you think of like muscle fibers, those are long and thin, and a collagen fiber is somewhat similar, so it also links together with other collagen fibers, and that linkage is pretty important to create kind of density and to create a structural unit out of that collagen fiber. So essentially, they just removed the actual collagen fibers themselves are still intact. It's just the cross-link between the collagen fibers has been removed. And what they find is that when they inhibit MT1, which is the same exact thing that they did for figure 1, except in this situation, even though they're knocking down MT1, there's still cell migration. So there's still cancer cell migration. And even when they use a general inhibitor, what they find is, again, that you're still seeing cell migration. So this essentially implies that if you change the collagen matrix in terms of somehow disrupting its actual natural system, the way that it works, the way that it's cross-linked, the way that it's connected together, if you disrupt that in some way, that the cancer cells are able to move without MT1 degradation system. So they're able to migrate through the collagen system and move through those pores that are created. And C is essentially just looking at both cell lines, again, that fibrosarcoma as well as the triple breast line or triple negative breast line. And again, they're finding that there are no differences, so that you're still seeing cell migration with the MT1 knockdown as well as with the general inhibitor. In figure 3, what they're trying to do is make it more applicable to humans. So far they've used kind of a generic collagen matrix, but in figure 3, what they want to do is use what's called an ex vivo system. So it's not as good as an in vivo system. An in vivo system is where you're going to be using the actual animal or the actual human being and implanting it in them or just watching it within a living system. But an ex vivo is using essentially culture or tissue from a living organism and then seeing what's going to happen if you use your intervention method on that tissue, on that more lifelike tissue. And in this situation, they again wanted to see what would happen if they tried to inhibit the MT1s, but specifically on collagen that comes from mammary glands, so right where you would find breast cancer. And what they found is that if they try knock down MT1, that they yet again see a decrease in the migration of those cancer cells. So if they can knock those MT1s down, then there's less migration of those cells. Now, again, after 3 days, they also showed that if they knock it down and then they reintroduce the MT1s, the expression of those MT1s, and allow the cancer cells to rebuild more of those MT1s, then the cancer cells are allowed to progress outwards. And in terms of just the bar graphs, I mean, B just shows the human mammary gland there, but it's obviously shows that if you knock down or try to knock down the MMPs 1 and 2, so the secretory MMPs, the metalloproteinases, then you'll see no effect. So you see normal cell migration. And if you knock down the MT1s again, then of course you're going to knock it down. So that leads to no cell migration. And if you rescue it, then you reintroduce cell migration. Pretty self-explanatory. And then they also did it on mouse mammary gland, but this one they separated out the MMP1 and the MMP2, so they didn't do it simultaneously. They did, but just later on. And again, same results. So the only difference being that MT1, if you knock that down, that membrane-bound MMP metalloproteinase leads to knocking down the migration of those cells. Okay, for the final figure, which is figure 4, they looked at the HT1080 cells, which are those fibrosarcoma cells, and they also looked at PMN cells, which are polymorphonucleic neutrophils. And those PMN cells, and I'm going to refer them as PMN or neutrophils, are innate immune cells, or they're just immune cells. They're found in our immune system. So those are considered healthy cells. They're helpful cells. And they wanted to compare this cancer line and see what kind of an impact does that cancer have on the actual collagen matrix itself. So do you see any sort of disturbances when it's burrowing through that collagen matrix? Are you seeing any differences? So in A, they're using two different microscopes. They're using a light microscope, which is essentially what you would normally use. Like if you go to any science lab, even in high school, and put something under the microscope, that would typically be a light microscope. So you can just distinguish what is a cell and what is not a cell. However, they also used a confocal microscope, which is a far more sophisticated method of doing microscopy. And what they find is that there are holes that are found in the HT1080 cell matrix. So in the collagen matrix, you find these pretty big holes or pores that the cell is degrading into and then kind of going through. So it's a really interesting image that you're looking at here, because it's really showing how those metalloproteinases really degrade into that collagen to allow that migration to occur. Now, on the other side, they have the PMN cells, and there you're getting migration as well, but there's no movement of the actual extracellular matrix. So you're not getting any holes, you're not getting any pores, you're not getting any of that stuff. It's completely intact. And yet those PMN cells are able to move through that matrix, which is a really fascinating mechanism if you think about it, because the cancer cells, while they are able to do a similar process, or theoretically they are able to do a similar process, the PMN cells are so well designed that they can change their morphology and really collapse in on themselves and expand outwards to be able to move through these collagen fibers. And then if you look at the bar graphs, they use different protease inhibitors. And of course, if you inhibit the proteases, then you'll see no cancer cell migration, which makes sense because, again, you're inhibiting that MT1. And if you use the same system, if you use those protease inhibitors on the PMN cells, because they are not limited by the MT1s, that in that situation, then of course you're not going to see any sort of impairment of the cell migration. So the PMN cells, the T cells, and the monocytes are completely uninhibited and have no problems with it, even if you add an inhibitor to them, because that inhibitor is not going to block anything that they actually use to be able to migrate through, which is not the case for the cancer cells. And then they've got a measure of hydroxyproline. I did mention earlier that the collagen fibers are made up of 3 different molecules. One of those molecules is hydroxyproline. I believe proline is another one, and I don't know what the other one is, but they decided to look at hydroxyproline. And you'll find, of course, as it makes sense, if you're degrading the collagen from its structural components down to its constituent components, its different amino acids, hydroxyproline being one of those constituent molecules, then of course you're going to be freeing a lot of that hydroxyproline. So you can essentially just measure how much hydroxyproline is in that environment, and you'll find that with the cancer cells specifically, with that degradation, you are seeing higher levels of freed up hydroxyproline, which is a great way to determine if the cancer cells are actually degrading the collagen itself. And of course, you don't see an elevation of that hydroxyproline when it comes to the PMN cells, T cells, and monocytes, because they aren't going through that degradatory pathway. And finally, for the last graph, they wanted to show what would happen if you plated these cancer cells in the same exact environment, so on a collagen matrix, and then you change the pH. So you're going to make it more acidic. So the first two, 5.5 and 6.5, are more acidic, 7.5 being more physiological, around that 7.45 range is physiological pH, and then an 8.5 being slightly more alkaline. And they supplied some images there for the microscopy in terms of what the collagen looks like at these pHs, and you will notice that there's a bit of a difference in terms of what it looks like. There are larger pores in the 5.5. In the more acidic situations, you're going to see collagen fibers kind of separate out a bit more, but it doesn't seem to have any sort of impact. You know, the different pHs do not seem to have an impact on the number of cells that are able to invade. So it's completely the same across the board, regardless of pH. But of course, if you add an inhibitor, which they do with the BB2516, then you see a vastly reduced level of migration. Now, this is an interesting point, and I find this graph specifically really interesting because you'd think that with these collagen fibers creating more space, having larger pores in the more acidic situation, that the cancer cells would be able to migrate without the use, even if you knock out, which they do with the BB2516, even if you knock out the MMPs, especially the MT1. And what they find is that no, the cells aren't able to. And I looked up another paper actually that did this exact same experiment, and their thought process was, and you can take it or leave it, but their thought process was that the collagen fibers are hypertrophying to a point where, although there are larger pores, the cancer cells can't budge those fibers out of the way, so they're stuck in place because the collagen fibers themselves are hypertrophied to a point where they've gotten much thicker and much more robust. So that makes it unable, even with the cancer cell having more room to move, it still can't move through that collagen matrix. So that's kind of the thought process. If that's the case or not, it's just a theory, it's just a hypothesis, so who knows. But anyways, that's what they have for the paper and what they have for their figures. And with that said, let's jump to the conclusion and the outro. So that's everything that the paper has to offer. Definitely let me know your thoughts and what you thought about this paper and what conclusions you would draw from the information that we covered. But in terms of my conclusion, I think we can certainly say that certain cancer cells, at least the ones that they investigated, led to migration through MT1 specific dependent on MMPs. So looking at not just any MMPs, but looking at the metalloproteinases that are specific to the membrane and how those are integral for the movement of cancer. Now, if you knock those out, you can still get migration of the cancer cells. It's just that the environment has to be conducive for that actual migration. So if you have linked collagen that would need to be degraded to move through, then cancer would be inhibited by the fact that it doesn't have its MT1 specific MMPs, but certainly the other MMPs most likely have some use as well in terms of migration. So it's interesting. You can certainly say that it has a dependent system and it has an independent system, but just because it has an independent system doesn't mean that it doesn't have backup systems, other ways that it can move through and migrate through that are, well, MMP independent, but certainly still dependent on other systems. So we don't know that, but we do know that MMPs can be useful in terms of moving through and migrating through that collagen. Unfortunately for us as humans, the cancer will actually create holes and degrade through our collagen and cause all kinds of issues in those ways. So it's certainly not a beneficial situation for us. But the paper is certainly interesting, and it does shed some light in terms of how we can understand cancer cell migration. And we even learned a little bit about human immune cell migration, which is pretty remarkable in its own right. All right, well, with that said, hopefully you enjoyed the video. Hopefully you found the video informative. And with that said, hopefully I have the pleasure of speaking with you next time. Have a good one. See ya.